This poster represents the three stages of cellular respiration. The first stage of cellular respiration, shown here, is glycolysis. In glycolysis, which occurs outside of the mitochondria, this region shown here is the mitochondria, and this region out here is outside of the mitochondria. One glucose is broken down, broken in half into two 3-carbon sugars by ATP, which phosphorylize the glucose and leave behind 2 ADP. Then, through enzymatic reactions, the 3-carbon sugars are used to form ATP and NADH, and also 2 pyruvates, which are 3-carbon sugars. Pyruvates are then broken down into CO2, and the 2 carbons that remain from each enter the mitochondria and are used for coenzyme A, which is an important participant in the Krebs cycle, the second stage of cellular respiration. In the Krebs cycle, the carbons from coenzyme A are carried around the cycle, forming first citrate and then oxaloacetate after 2 of the carbons are actually 4 of the carbons are given up as waste from CO2. And that's 4 carbons total from the both pyruvates, 2 carbons from each pyruvate. That is a waste product. The important products for the next stage of cellular respiration are 2 FADH2, 8 NADH, and 2 ATP. The ATP can immediately be used for cellular processes, but the NADH and the FADH2 are used to react with the first transport protein in the electron transport chain, which is the third stage in cellular respiration, effectively giving up their electrons and their hydrogens to become FAD plus and NAD plus. Their electrons travel down the transport chain, and this triggers the transport proteins to pump the H plus ions, which are essentially just protons, through the inner mitochondrial membrane and into the area between the outer mitochondrial membrane and the inner mitochondrial membrane. This creates a concentration gradient for the H plus to diffuse back into the mitochondrial matrix, and the path for them to do this is ATP synthase. And as they diffuse through ATP synthase, it causes ATP synthase to react ADP, converting it into ATP by adding a phosphorus ion. Through the ATP synthase, 34 ATP are made, adding to the 4 ATP made from glycolysis and Krebs cycle combined, to make a total of 38 ATP from the process of cellular respiration.
And here we can see a light reaction happening with photosynthesis. And here is photosystem number 2, P680, surrounded by chlorophylls, which attract light. This funnels the light into the photosystem, which is basically a big sacrificial altar. And H2O is ripped apart into 2 hydrogen ions and 2 electrons. These electrons are then sent up to the primary electron acceptor, and then, through the electron transfer chain, come to photosystem number 1. Again, the same deal with the chlorophylls, which attract light, and what happens is the electrons get re-energized, so they're sent to another primary electron acceptor. Then, through another electron transport chain, they're sent to NADP reductase enzyme. What this allows to happen is NADP plus 2 hydrogen ions plus 2 electrons creates NADPH, effectively known as an electron carrier.
Hello and welcome to the Calvin cycle. here we have our Rubisco enzyme, which, when CO2 molecules come into it, make a 3-phosphoglycerate, which is comprised of 3 carbon atoms and 1 phosphate. Then ATP phosphorylates into ADP, thus giving 1 phosphate to the now 3-biphosphogycerate, which is right here, which has 2 phosphates. Then the electron carrier from our light reactions, NADPH, gives its electrons an energy, turns into NADP+, and sends 1 of the 6 biphosphogycerates out of the system, leaving 5 G3Ps remaining, which then, when acted on by ATP, which then becomes ADP, gain a phosphate and reorganize so that they are a 5-carbon molecule with 2 phosphates called RUBP, which then recycles through the process. Each time 1 G3P is lost, which is half of a glucose molecule.